have the same again. We've got Lucy, who is our sort of tech wizard, who's going to help us out for any of those things. We've also got Rob and Claire, two other vets from the practice, and we've got Katie Mackenzie as well from SIVA Animal Health. She's a ruminant veterinary advisor, and they've kindly sponsored us this time. So we're going to sort of work on the sort of tech side of things, hopefully tonight, not run into any problems, but we've spread ourselves out rather than from doing it all the same place. Now there'll be questions and discussion at the end, hopefully, but if there's any questions that come up as we're going, stick those up on the chat board, and we can hopefully gather those towards the end or answer them as we go along. Cheers, Lucy. So the plan for the evening, bit of an intro. Why have we chosen integrate parasite control as a subject? Where do we think it fits in? How do we think it fits in with the whole subject of regenerative agriculture, and where can we help? Kitie's going to tell us a little bit. She's got a very unique sort of view. Kitie was a practicing vet for a long time and is now in industry. So she brings a great deal to the discussion, really, having that view that straddles both the industry side of things and practically. Rob's going to talk about what we're already doing, because this is an area of the practice field we can help. Claire's going to talk a little bit more about what we can do to advance our sort of practices in this area. And then we're going to chat a bit, have some discussion, some questions, which we got a really good sort of interactive session on last time. And we want to hear what you guys are doing as we go along. So keep those questions coming. And the idea is to finish by 9. That's quite a nice thing with Zoom meetings, I feel. We're not going to have one of my sort of marathon 5-hour meetings. Cheers, Lucy. So initially you might think, well, how do these two things fit together? But we talked about regenerative agriculture last time, and essentially we're looking at enhancing our environment and our farming businesses, and improving things year on year to everybody's benefit, those of you guys as farmers, public, animals. And so with integrated parasite control, we're aiming to be more targeted than conventional control, where we might just use sort of diarised calendar approaches. And we want to talk a bit more about simply what product do we use and when. It's not as simple as that, and it's not as simple as don't just use wormers. So really we want to be led by diagnostics and evidence-based, and with a view really all the time on the environmental, the animal health side of things, and then of course with financial and production benefits for you guys, or there wouldn't be that sort of symbiotic relationship. So we think that these two elements, regenerative agriculture and integrated parasite control, share a lot of things. So of course they both want to enhance animal health and performance, that goes without saying. I've put in there about achieving sustainable animal production. Clearly regenerative agriculture is simply more than being sustainable. We're pushing it onto the next level. We want to achieve more from less in terms of feed utilization, make the most of grazing, get less waste, which in this case is talking about parasite burdens. We want to see financial benefits because we want our businesses to be more sustainable and the livestock and agriculture industries to be more sustainable and to help solve some of the issues we've got facing us. Of course, we want to look at improving our soil ecology. We know that some of the products we have will affect that, and this is an area that is key to regenerative agriculture that we want to expand on in other meetings. We want to minimize our interventions, that makes sense, and wormers are a big part of that. But if we can minimize the underlying challenge, then that will help hugely. And we'll look a bit more about the targeted treatments and make sure they work. And the big thing with both of these is year on year, we want things to improve. We want things to be easier and easier. And so next year, our lives are a lot easier and the job is a lot easier. Thank you, Lucy. So really the big thing about moving from a conventional approach in parasite control to a more targeted and one requires a change in mindset. And you guys will all be okay with that. The fact you're all here means you're open to a change in mindset. The fact you're considering regenerative agriculture practices means you're open to a change in mindset. But there are some challenges there, and there's some challenges specifically around some of the principles of regenerative agriculture and how we integrate good parasite control. For example, one of the principles is to minimize soil disturbance. That makes sense. But equally, one of the things that can help us to manage parasite burdens is new lays and rotation. So there is a bit of a balance there that we need to talk about. Thank you, Lucy. So the conventional approach, you'll all know about that. It's very simplistic way. People would use calendar based approaches, worming every 5 weeks. And of course, it requires a bit less thought. You often think, well, it's worked in the past, it will work going forward. And it gives you a bit of a false sense of security. The difficulty with that approach is we're often kicking problems down the lane a bit, and we're making issues for ourselves later on. Whereas a targeted approach, it is more challenging. It requires more engagement, more thought. And a big part of that is dealing with a situation that changes year on year. It will be different between farms. It'll be different between years. And it's even different between fields within the same farm. So to make this work, we have to get really engaged with it and be prepared to change our plan a little bit. But with some good planning that we'll talk about tonight, we can do that. Cheers, Lucy. So this next slide is just a little bit of a reminder about some of the pressures on us. So this is taken from SCOPS, which is sheep-based control of parasites, but the same thing applies to cattle. So we can see on this graph that as we move towards greater resistance, and so less of the worms being killed by a product, we get a decrease in animal performance, whether that be cattle or sheep, whether it be growth, immune function, or milk yield. And so really, we have to understand this because it's a big issue in the industry, but it definitely interferes and contributes towards our challenges when we come to parasite control. And the thing to remember about this is it's the worms, the parasites that are resistant and not the animals. So simply animals go in, those parasites are still on your farm and are still resistant. The next slide is just a reminder of our worm groups and how the resistance plays into those. This is a slide from Fiona Lovett and her Flock Health Limited company, and you can see there that actually a lot of our existing group 1, 2 and 3 wormers, there's very widespread resistance to there. So even removing a regenerative agriculture angle, there is still clearly a big drive for us to manage our parasite burdens more holistically. So we talked a bit about targeted diagnostics and how this approach really does need to be diagnostic led. And here we just have a few of those examples. Clinical signs, I'm sure you all use those, but we can go beyond that. We can look at antibody levels and exposure within animals. Postmortems are incredibly valuable. We can do those on farm. We can even get to the point where you guys are doing some rough ones on farm. Worm egg counts. It'd be good to know in the chat how many of you are using these different diagnostics. I would hope a lot are using worm egg counts, but we can look at liver enzymes as evidence of migration of immature fluke. We can look at antigens within the feces to get an idea of fluke infection before we get to the egg stage. And then if you're selling direct to abattoirs, it's very useful to get the abattoir reports back. We're going to talk a little bit about proenzyme levels. Claire might mention pepsinogen, and we talk a lot in the sheep side of things and the pre-weaning, certainly in cattle, about daily live weight gains and how we can predict problems if these animals fall off the trend, either individuals or groups. And I know Claire is going to mention some of the parasite forecasts, which are incredibly useful to help us predict problems. So these are some alternative strategies, and we might wish to get into a bit more of a discussion on these later on. It'd be really interesting to know what you guys are doing from this sort of little selection. So a big part of this is managing the exposure of the environment, the levels in the environment of parasites, so we can completely negate the need for chemical interventions if we're able to limit the amount of exposure animals are getting to parasites on the ground. And so things like rotational and shared grazing, where we use the different species of cattle and sheep to dilute worms, and alternative forages. We know about chicory and how using chicory in our grazing management can actually act as a really, really effective anti-parasite agent, decreasing burdens by 40–50%. And using adult animals as hoovers In the majority of cases, adult animals are far less susceptible to parasite burdens, excluding liver fluke and to a certain extent lungworm. And so by using those animals at sensible times in our production cycle, we can clean up pastures from the parasites that may affect some of our more vulnerable populations of animals. And anyone who knows me will know that I believe that nutrition tends to be at the center of everything. And so there's ways that we can manipulate nutrition to convey greater resilience to parasite infections to our animals as well. And then when we do need to use treatments, if we're able to use very targeted treatments using the correct product at the right time to the right animals, we are able to really have a great effect on the effect of any parasite burdens. And there's just a couple of slides to illustrate some of those points. So what is very, very useful in integrated parasite control is at the start of the season, before we turn out, is to look at all of our grazing areas and to grade them as low, medium or high risk, depending on their grazing history or the crops that we may have had in the rotation beforehand, or what makes up the lays in those pastures. And by doing that, we can then assign our different areas of risk to different classes of animals at different times. So that's just taking that on to the next level and really should form part of all of our integrated parasite control plans. And then finally, this is some old work from the early 90s, but then has been built on with various other studies that has really proved it quite nicely. This was just a small project in Harper Adams, but what it showed is that animals on a higher plane of nutrition, specifically with respect to protein, were far more resilient to worm infections. Their excretion of worm eggs was far, far less. In this case, it happened to be used around lambing, who always suffer a decrease in immunity, but the same thing applies to other animals and other ages of stock. And the important thing to note here is the low protein diet. That wasn't a protein deficient diet. That still met all of those animals' needs, but by increasing the protein level, we were able, as you can see by the level blue line, we were able to achieve a much greater resilience to worm infections there. So what I've tried to do there is just take two of those examples and show them in a little bit more detail to show that we have a lot of other options available to us. So I'm going to hand over to Kaiti. I can see there's some questions coming up on the side there and examples of what people are using. So we'll keep an eye on those and talk about them afterwards, but I'll hand over to Kaiti.
Hi. Yep, thanks, Dan. Thanks for a very nice introduction as well, and nice to be here with you all. My random question of the evening was, am I a unicorn? So I am a vet who works for a pharmaceutical company, and I'm going to talk to you tonight a little bit how you can use less product, which is possibly a little illogical. So when we look at agriculture, obviously, I think we're all on this, you're on this meeting. We need to provide food that is safe to eat and affordable and produced in a sustainable and regenerative way. And the demands for meat and protein and milk are massive, about twofold increase by the end of 2050. So we've got a big job out there to try and get this right. So I'm lucky enough to work for a fairly small French-owned company, and we try to support agriculture. So they're very keen on animal welfare, and we do a lot of work in trying to preserve agrobiodiversity, as well as selling some product. But we're very keen to work with you guys, which is why I'm here tonight. Next slide, please. So I think we can all agree parasites are a major challenge to ruminant health and production, depending on whichever species. I think we've probably got everyone here, dairy, beef and sheep. And we need to balance this in terms of using enough product, but not too much. And particularly when we look at the anthelmintics, so the worming products, especially the MLs, so your group 3, your ivermectins, your moxidectins, those sort of products, they do have an environmental impact. So we've got a few challenges in trying to get this integrated parasite control to control the parasites without causing too much more harm. Thank you. So goes to the old saying we all have, use as much as necessary, but as little as possible. So we need to worm the animals that do need worming. And this goes back to our butterfly effect, which should fly in next. So little movements anywhere have big effects elsewhere. So there's my little butterfly. Thanks. So what we can do in terms of this, when we look at worming, is look at two sort of different things. So we can look at targeted treatments, where we might do the whole flock or the herd or group, but done on the basis, as Dan just said, of known risk factors or parameters that we know what we're dealing with. We're not just doing it because it's the 4th of January or July. We're doing it because we know there's a risk, or we know the disease is there, or the egg counts are increasing. And what the other thing I really want to talk a little bit about tonight was targeted selective treatment. And this is where within a grazing group of whatever animals we're talking about, we try and treat very specific animals, that the animals that need the treatment. It goes back to as well the 80-20 rule. There's an argument that 80% of the parasites are in 20% of the animals. So it's trying to pick those animals out. And we look at things like fecal egg counts, daily live weight gains, milk yields, body condition scores. So what we'll do for the next 5 minutes, hopefully, is just look at what we might do and what impact we might have. So thanks. So yeah, hopefully we need to minimize growth rates, changes. We want to make sure they grow as well as they can. And I'm going to show you a little bit why that's important. And we need to obviously balance removing this burden. And so can we use TST? So just the next couple of animations would be great. This is just some work that was done recently in 2013 in the UK. 4 different treatment groups. So you can see them there, monthly, treated every 4 weeks. Strategic was based on risk factors. TST, so very targeted, based on daily live weight gain changes, and clinical signs was increased fecal egg counts and or evidence. And what they did find was a significant treatment effect. So this leaving it until we get clinical signs was potentially having an impact. So we're getting decrease in growth rate, and actually did end up with being an increase in carbon dioxide emissions. So there is a balancing act to get it right. And hopefully that paper just demonstrates that a little bit. So the sort of conclusion would be that we can contribute to a reduction in greenhouse gas emissions, which helps to try and reduce the carbon footprint if we do things right. So when we look at TST, we're going to look at decreased use of anthelmintics, as alien as that is, less product to be excreted onto the pasture maybe, maximize the growth to treat the affected animals. But we do need to make sure that we're not having negative health or welfare indications when we do this, and try and minimize the environmental and carbon footprint. Just a little bit on dung beetles. I know you're going to hear a lot more about this, as people on here are much more knowledgeable than me about it, but I just thought this was quite interesting. They looked at whether the amount of drug that came out in the feces or the percentage of animals had more impact on the dung beetle selection that was available. And it turned out that if you TST was a lot better at maintaining the variation compared to effective drug concentration. So we need to be very specific with which animals we treat. So how are we going to do this? Let's start with our friends, the sheep. So in the small ruminant world, we are definitely much better. We've done a lot more work, and we've been a lot more proactive, I think, as an industry on it. So we know can look at daily live weight change. We look at body condition scores. We can look at DAG scoring. We can look at mob fecal egg counts. And from that, we can lots of different things. So there's been a lot of work done looking at treatment of ewes at lambing. And actually, a lot of work would suggest that there's no effect on the long term. Yes, you might have a short term impact, but really, if you're going to do this treatment, you really need to make sure you are treating animals that actually have a burden, because in the long run, it didn't appear to have a massive significant difference. So again, to use as TST, so targeted selective treatment, if you look at daily live weight gain and or breech soiling, in this work done here, targeting just specific animals with either breech soiling or change in daily live weight gain, the TST had no negative impact, but it did reduce the use of wormers by 50%. So everything was still growing, everything was still doing what it should be doing, but we're using half the amount of wormers that we were before. The happy factor just made me chuckle. This is a clever computer program, modeling program, and you put in growth and grass and type of lamb, and it works out how much they should be able to grow. And that's the happy factor. And then what they do within that is, if lambs are not making about 2/3 of what they should be able to do, they would then be eligible for targeted selective treatment, so this TST. And what they discovered was that doing it this way, there was very negligible negative results. So I think in the sheep world, we have things we can do. Moving on to beef, cows and calves, really a bit more of a challenge. So I would say suckled calves probably don't need to be treated when they're on their mother. So if Lucy can just click in the next couple for me. Sorry, I forgot I'd animated all these. Thank you. So suckler calves rarely need to be done. First season grazing dairy calves, we can use TST, certainly. There's work out there. As Dan mentioned earlier, we should be a little bit careful with lungworm, particularly in our dairy heifers or on farms, so we know it is. I would argue we just need to vaccinate, and that's not even made by my company. I think vaccination for lungworm is a really good idea. Pepsinogen is quite useful. You can actually use this either halfway through the season or at the end. The reason we can measure this is that the Osteotargia, which is the main stomach worm in cattle, it infects the abomasum, so the stomach. It actually damages the gastric glands. So it burrows in through the gastric glands and comes out. So it changes the pH in the stomach, which impacts on digestion and appetite. But it also means that pepsinogen, there's higher levels of pepsinogen because it's not converted. So if you were midway through your season and we did some pooled pepsinogen samples, that might indicate that actually there's been more osteotargia than you're thinking there is, and actually some anthelmintic treatment might be necessary, or it can be used maybe at the end of your first grazing season to assess how well your protocol's been working in terms of grazing management. Daily live weight gain in grazing calves, either dairy or beef, older beef animals. I think you need to look at as a percentage of change maybe for the animals, because there's so many other factors, genetics, etc. Nutrition, going back to Dan's favourite subject, will have a massive impact on daily live weight gain, so it's important. I think, as Dan mentioned as well, antibodies can be useful. You can use them for osteotargia in dairy cattle. Again, it's monitoring. One snapshot does not give you any answers, I'm afraid. You have to be doing these things quite consistently. And I would say that fecal egg counts in adult cattle are challenging to be of any use, will be my polite term for it. Looking specifically at dairy then, just a few ideas. This is some very recently published work. So we know that parasites can control, have quite a big impact across a herd. Milk yield's probably the most obvious and actually something that we can measure to see if we're getting an effect. Parasites can also impact on reproductive function as well. So on this study here, it was done in France, funnily enough, for a French-owned company. We did a lot of cattle, but the great answer was that, so any herds that had a moderate to high level of grazing, so this meant they were grazing for, grazed grass was a significant proportion of their diet during the season. On these herds, just targeting those herds, we could gain up to 1 kilo a day on their milk yield following treatment with an injectable epinephrine. Time effective contact, I think Rob is going to come back to, but this is quite an important concept. So we talk about worms getting resistant, and that is really important, and there's more and more of it about. But cattle also develop a degree of immunity or resilience, depends how you want to describe it. And against the two main gutworms, the Cuperia, they gain a fairly quick immunity, and that seems to last. Osteotargia, they actually need at least 8 months of grazing, so probably 2 full grazing seasons, and even then, heifers born at a strange time of year, a different time of year, may not get that effective contact. And this actually impacts quite significantly on how they then respond to meeting the parasites later on. So if we added to the fact these herds had high levels of grazed grass, if they also had low time of effective contact, that boosted the response even further to a worming. And bulk milk ODR, so that's just a measure of the antibodies to osteotargia in the bulk milk, needs to be monitored, but in these herds, the higher levels again got it. What we tend to find is very high or very low bulk milks respond well to worming. In between's a bit variable. When we look at the cows within these herds, interestingly, they don't need to be low yielding. Apologies, got to change that. They need to be lower yielding. So I think we used to always think that the high yielding cows would be the ones that were at risk. But I think what we learn now is that the girls that are really performing well probably don't have a parasite burden. But you might have girls that should be just giving that little bit more. So they're just not quite up to speed. So those cows and less than 200 days in milk, they're the cows to pick. We've got a program sorted to pull this information out of one program, and I am waiting, hopefully shouldn't be long, till I have the data for the other program that Rob wants. So these guys can take this program and actually pull these cows out specifically on your dairy herd. And that was conclusions are next, I think, which hopefully will fit in. So work with your lovely vets. What an array. How do you not work with these lovely people? So know and understand your burden. Look at managing responsibly. Consider, in terms of dung beetles, how long we can keep animals away from soil, how quickly these products get degraded in the feces or in the slurry. Grazing management, yep. Effective production is what we want. And definitely think about a whole herd, flock, farm plan. You need to think about everything, all your parasites in one big place. And these lovely people from LLM are the people to help you with that. So that's my sponsors bit over. So thank you very much.
Thank you, Katie. That was great. So I've been asked to talk a little bit about what we've been doing as a practice already. And I think it's important to say it's really important for us that we want to make sure we've got all the services that are tailored to the likes of everyone who's on this call. But also it's really important for us to engage and offer services to all of our clients who perhaps have systems that preclude sort of the full embracing of the principles of regenerative agriculture. So in other words, helping people move from one end of a scale up it, towards regeneration. So a good place to start is soil health, and that previous slide was just a bit about. Thanks, Lucy. One of the principles, if you like, is this incredible fact that half and more sometimes of the carbon, the sugars, the carbohydrates that a plant generates through photosynthesis are actually pumped out through the roots into the soil, which you might think is a waste. And this then forms the basis of soil health, of the food web. And it was, I thought I'd start off with this slide that Niels Corfield's, I think, drawn, because it came up in the discussion in the first meeting we had, and about mycorrhizal fungi. So bacteria and then mycorrhizal fungi, that forms the basis of everything to do with soil. Thanks, Lucy. Next slide, please. And the importance of that food web that builds on top of that. And as Katie pointed out, ivermectin knocks out huge swathes of that fundamental web. It's an incredible product, but the question is, should we be using it as much as it is being used at the moment? Next slide, please, Lucy. So one of the things we've piloted this year is making use of a soil health app called Soil Mentor, and it basically allows you or us, the farmer and the vets, to do free soil testing on farm. So I'm sure many people out there will be testing pH and chemical analysis, but this anyone can do with a boot and a spade. Although what we found is a lot of our clients are too busy. So this is a way through our vet techs to make it hassle-free and start that process of engagement and looking at things like soil structure and how that connects with the life in the soil, like earthworms and dung beetles. And tonight, I won't spend a lot of time talking about dung beetles, although I'd love to. There is just to say that there's so much variety out there. It's not just one species, but they're struggling, and we've lost some species already. But these guys bring benefits to the cattle industry that amount to 367 million a year in the UK. They're a keystone species, and it's kind of been a useful tool to help engage everyone in what we're doing as a vet practice. Next slide, please, Lucy. So where did we start? We actually made some use of some SEVA funding to get out there. It's tough for farm vets sometimes. To move into this space where there's free advice and products are sold, we can't always compete with the pricing that's out there. So it's one reason many practices haven't been as involved as we might have been over the years. So that was really useful to get in and find out what's happening. And the pilot that we've done this year, this sort of slide illustrates one of the most common practices we see probably in dairy herds, which is that sort of calendar timed pour on use of an ml or ivermectin, and then possibly more when they come in as well to hit the fluke, but it might also include an ivermectin. So obviously there's big impacts of that. And I'll move on to the next slide again, please, Lucy. So yeah, the first has been mentioned tonight already, the potential for resistance. So a lot of farms have been using the same protocol for years and years and years, and if it's always the same chemical, then the likelihood of building the parasites ending up resistance is very, very high. And also, the pilot has shown, and I'll come to that in a second, but there's a lot of unnecessary treatments being used. So 920 quid was the figure on the previous slide for that farm, sort of 150 cow dairy. And by testing this year, we've managed to not treat on many farms. So there's a saving of money there. And then, as Kaiti was talking about, that time of effective contact is so important. And a lot of farms will be ending up not generating immunity in their young stock. So by the time they hit the adult herd where they might be productive, that's actually potentially the first time they have to deal with possibly lungworm, possibly ostatagia, and actually it's hitting milk production. And then the last sort of big area is often unthought of, which is the environmental benefits of beneficial organisms like the dung beetle. Thanks, Lucy. So the solutions from the sort of treatment and vet perspective, start a nice building block is vaccination, because and Claire's going to come on to this, it's very difficult to test for it, so it's a really, really good safety net. And then, as Dan and Kaiti both alluded to, rather than treating preventatively, we go in and we monitor what's happening using fecal egg counting and other techniques, and only using treatments as and when are needed. And if we can, we will rotate those products and try and avoid the use of the more environmentally impactful ones like ivermectin. And then, when they come in, we'll look at fluke as well. So this is a bit of a fag packet calculation and graph showing the outcomes of the intervention, and going and talking about parasite control on this group of around 20 farms. It will be written up in a little bit more detail, but starting from the left, we had no farms who were enrolled that were already making use of fecal egg counting. And through this season, 94% had done at least some, and 69% were actually doing what I've just talked about, which is diagnostic-led treatments. So really only treating when it was necessary, which I felt is a really good outcome. And then one of the other things, what percentage were using ivermectins, and that was almost all, and that could be for 10, 20 years. And at the end of this season, following the pilot, that's roughly halved. So half of them had stopped using them at all, managed to stop using them this season at least. And of those half that still use some, pretty much all of them managed to reduce the amount they used. And we even managed to jump from, we had 37% of the farms involved managed to go the whole grazing season without needing a wormer treatment, which was incredible. And the last point, quite a good number have either used vaccination or plan to use it next season. And actually, of the farms that have needed to treat, a lot of them has been in unvaccinated herds, and it's been because of lungworm. Okay, thanks, Lucy. So that's kind of talking about the treatments, but within regenerative agriculture, there are a whole host of other ways of managing and minimizing the impact of parasites. So it's certainly an area that I would love to get into the discussion, because some of you guys in this group, I think I saw Bruce Thompson, I had a chat with him recently about some amazing stuff he's done, but you've got, yes, I think someone mentioned chicory, herbal lays, you've got condensed tannins in willow, all sorts of methods to to reduce the need for wormer, other than just relying on treatments. So we'll come back to that hopefully. Thanks, Lucy. Yeah. And then coming back towards my favorite topic, the dung beetle, finding ways and means to engage our vets, our vet techs, our whole team at the practice, but as well as our farmers, has been critical. So we've spent a lot of time this year and invested in equipment and in systems and in documents to help get people out there doing it. But really, next slide, please. It's the dung beetle, and this is, I'm not going to go into it in a lot of detail. I point you towards webinars that are out there. So Sally Ann Spence and Bruce Thompson did a brilliant one at the Northern Real Farmers Conference, so go and find that if you can. And the Mans and Carbon Calling did a great one earlier in the year, and again I think Sally Ann did one on with the PFLA. But these are some of the areas that they generate their value to a farm, and so yeah, that's where that figure of 367 million comes from. Next slide, please, Lucy. Yeah. And I'm just going to focus on one small part of that, which is fly control as a principle, to sort of illustrate one of the things they do. So if you look at the little dung beetle here on the bottom right, there's a little mite attached to the back leg, and that guy is a phoretic mite, and it predates the maggots, fly larvae, basically. And all they do is they harmlessly cling to the legs or abdomens of the dung beetle, and as the dung beetle flies between pats, spreads those mites, and they actively act as a biological control to keep fly populations down, which is quite an amazing thing to see when you see like tens of mites clinging to these dung beetles. Thanks, Lucy. So this, and it brings me to the second part of, or third part of what we've been doing, which is trying to deliver surveys for farms to look at both the abundance and the diversity of species in the farm. So this is an example of a farm that had really good diversity and abundance. And I won't go through all the species, but not everyone, there's plenty of farms I'm going to that have been using ivermectins for many, many years, that I find, I've got some that I've found none, and most you'll find one or two different species at least, but this guy had a lot, and this was just on one visit. And this illustration just shows, well, for me, some of the beauty of what you find down there in the cow pats. Everything, different colors, sizes, shapes, all sorts, and you slowly build, you know, your interest, and they're incredible little guys. Next slide, please, Lucy. But it isn't just the dung beetles. There are huge variety of bugs that live in there. Some of them are predators, and like I've just described about the parasite, the phoretic mites, and some of them eat muck, and they get rid of the muck as well, and that has a benefit for the soil and for allowing the grazing to come up. Next slide, please. So here's an example from that same farm. 7 different species of, not species actually, groups of predator, from mites to flies to rove beetles, these sort of long thin things, clown beetles, soldier flies, and parasitic wasps. And for those of some of our clients will actually be using, you can actually buy parasitic wasps and spread them on your farm to help control fly populations, but they also occur naturally. So if we don't knock out these guys, they will be doing great work for us. So next slide, yeah. And then this is the coprophagic, so the bugs that eat the dung, other than dung beetles. Some of them look a lot like dung beetles, and on this farm there was too many to count, thousands. And what was so incredible was the fact that of all of the pats that I looked through, I only managed to find one fly maggot, and that's because of all the predators and the fact that a lot of the dung is being removed by all these other guys. Next slide, please.
Rob,
yeah.
Can I just raise something? We had one of the questions on the chat before, which was about controlling maggots without knocking out insects that feed on dung. In the specific example, it's dung flies really that some of these products are quite dangerous against. But you've sort of answered that, haven't you, in your roundabout way, and that by allowing these invertebrates to populate, then we have far less of an issue with nuisance flies and maggots and that type of thing as well. So it follows on. Would that be fair?
Absolutely, and it's kind of, that's the kind of point of the point of all this, is by it's demonstrated really graphically to me the power of on-farm ecology to help balance and control some of the things we're otherwise having to treat for. And the last slide here is talking about the other wildlife that comes in on the back of all that life. Again, this farm for most of September, I was writing it in the reports at this slide that, oh, well, it's, you know, the swallows, the house martins, they've flown back to Africa. But the farm I've just talked about, with all of that life, still had scores and scores of house martins just flying around the field, even into mid-October, which sort of blew my mind. I don't know how everyone else has seen them on their farms, but they seem to be hanging around this farm a lot longer. Okay, next slide. Yeah, just to mention that if there's time and the inclination, I've got a little video we can share at the end. So that's it from me. Thanks, everyone.
Hi. Oh, sorry, go on. Sorry, Dan.
Sorry, Claire, I was just going to introduce you, but you far away.
You can introduce me if you want.
It would have been offensive, so probably best to get you.
First off, sorry you guys can't see me. I'm actually in Wales at the moment, so I'm blaming it on that. It's not because I'm smoking or drinking a load of whiskey or anything. I just haven't got very good bandwidth. So basically, I just wanted to talk a little bit about diagnostics, things that we can do and that are available to us at the moment. I'm only going to talk really today about lungworm, gutworm and fluke. There are obviously lots of other parasites out there, coxy, crypto and flies being some of the other things that we see. We've had a little bit of a chat about flies, but we reckon we've probably got some more stuff for a whole other meeting there as well. So we'll go through these. As I say, there's lots of tools available, and knowing what to use and when is important. And one of the things I would say on here out of this list is I would never underestimate the value of a post-mortem. I think Katie mentioned it a little bit earlier, but if something dies on farm, often it is we see it as a waste, and often we don't want to look at it. But I think trying to use those animals to your advantage and getting your vets out to do a post-mortem can be really, really useful. So things like lungworm, liver fluke and gutworms are really easy to see often on post-mortems. So if you can use those animals, and then often it's, you know, I see it not as quite so much of a waste there as well. So Lucy, can you flick forward for me? So when we talk about gutworms, and this is my picture that I drew in the corner, there's a worse one to come up, and I did do A-level art, which may go some way to explaining why I am a vet and not an artist. But this is where life cycles become important. Now, if we understand how a parasite works and how its life cycle works, we can better understand how and when we need to control them. So the whole cycle of a gutworm in cattle and sheep are very similar in their gutworms. So the cycle takes about 21 days. So the cows ingest the larvae off the pasture. It goes through the cow, or the sheep comes out the other end in their muck, and then they have a development stage in the muck onto pasture, and it goes round again in a circle. Now that whole cycle takes about 21 days-ish in a cow and about 16 to 21 days in a sheep, so maybe slightly less there. And there's various things that we'll see with that. So I think we've all seen clinical signs. So that calf there on the left-hand side, they're the sort of clinical signs that we'll see. And you get similar in sheep as well. So you get this sort of failure to thrive. You get animals not putting on weight. You often get this quite starey coat. We often see emaciation, dehydration and diarrhea as well is a common sign that we see. And often we'll see it in groups, as a group rather than as an individual. If you have an individual animal group, it may be a sign of gutworm, but it's less likely we tend to see those signs across the board when we're talking about gutworms. So the problem is we don't want this calf on the left to be our starting point. So if we're seeing clinical signs of gutworm like this, something has gone wrong somewhere in our control planning. So what other things can we do? So Rob and I this year have done a hell of a lot of fecal egg counting, probably more so than I've done in my career. And going back to what Rob said earlier, it's often been an area we struggle to get included on, really. It's often, and I don't mind admitting this, it's been a part of the health plan that I often ask the farmer what they do, and I've gone along with it because they'll often buy their worms quite cheaply from trade, and it's something that it's kind of been a box ticking exercise. But this year we've got a lot more involved. So what a worm egg count will tell us is the approximate number of eggs that are contaminating the pasture. So we need to know sort of when we're going to start doing them. So what I will say, and again, sort of learning this year, is it will depend on your turnout date. So I wouldn't necessarily do the same timings that someone else is doing, just say, well, we'll start in April. Look at when your turnout date is, and then you want at least one cycle to go through the cow to come, or the sheep to come out, to be able to get a decent result. So I started doing them from about 4 to 6 weeks after turnout. If they had overwintering cattle, I did start sort of late March, I think early April, just to sort of give us a bit of an idea and try and start early enough that we'd start to see changes as they occurred. So it's important to remember that it isn't a reliable indicator of worm burden, but it is the number of eggs that are contaminating the pasture that you're actually looking at. And we should be interpreting these in the context of sort of risk assessment. So we need to be taking into account things like body condition, etc. And it can be a really useful time to actually have a look at your livestock when you're taking these samples. So what I've indicated, and actually this didn't start at the beginning of the year and things have changed as we've done it and I've got more involved with it, is when I've been out taking samples, I've started taking the percentage of mucky bums that I can see in the group. I've started to look at the percentage that are coughing. And then I've body condition scored about 10. And basically, if I've seen a difference in body condition score of over half a condition score between fecal egg counts, which have been 4 to 6 weeks apart, I've had a chat with the farmer about that and seen if there's anything else going on there before we just assume it's a parasitic burden. And the general status as well. So what is their coat looking like? Are they a bit starey? Do they look a bit dehydrated? All those kind of things as well. So in terms of doing a fecal egg count, I've tended to do pooled samples unless there's more of a concern. So each test should be based on at least 10 individual samples from a group. So you should take them at random and try not to pick and choose. And they need to be of fresh dung, so ideally less than 24 hours old. So if you're getting the cattle up or the sheep up for anything else to do, whether it's daily life weight gains or having a look at vaccinations, things like that as well, that's quite a good time to take those samples. So you can collect them into a glove or little pots like this chap is here, and just make sure that you expel as much air as you possibly can from those samples before you put a lid on it or before you tie your glove shut. Keep them in the fridge if they're not being done immediately, but ideally they want to be in the lab as soon as possible. So ideally within sort of 24 to 48 hours. And the reason that is because that life cycle continues on in that pat. So if you have eggs, they will turn into larvae and you won't get an accurate count over a period of a few days. So try and get them in as soon as you possibly can. I tend to say don't pool them yourself. So send them to your vet and then your vet can accurately weigh them out. If they're performing foot pooled ones, they can take the same amount off each animal, because although it might look the same amount, a sloppier sample might actually weigh less than a heavier sample, than a more solid sample, for example. And then if a farmer has been concerned about individual animals, I have said if you want to take a couple of individual samples off some cows that you're concerned about, we can just check their burdens as well, potentially. The best thing I've actually found is going in, if the cows are lying down, when they stand up, they'll drop muck. They tend to just have a crap as soon as they stand up. So that's been a quite useful time to take them. Or if you carry some pots or some bags when you're out sort of sheep herding or something and take a sample as they get up, that can be really useful. So the other thing I've put on there as well is the FECRT. Now that stands for Fecal Egg Count Reduction Test. And this is really important. And I've learnt my lesson with this one this year. So I didn't do one on a farm. They wormed some cattle with pour on wormer. I didn't actually know they'd worm them. And then I got a call to come and see them about 3 weeks, just short of 3 weeks later. And they didn't look very well. So it turned out they had been treated. It had actually rained an hour later. And I think it was just thought that they would probably be okay. So doing those fecal egg count reduction tests are really important about 2 weeks after you've used any treatment as well, to see if your treatment's worked. And you want to see about a 90% reduction or thereabouts in the amount of fecal egg counts you're seeing. So don't just assume it's worked. Always check to make sure it has, because it may not be performing as you hope. And that could be because of resistance, and it could be because of operator failure. It could be as simply as it rained. So performance monitoring. So again, that's things like your daily live weight gains as well, seeing how they change over time. If they're not performing as Katie was saying earlier, then potentially that's a point to make a decision about what you're going to do, and that targeted selective therapy as well. So using that on a certain proportion of those animals, if they're not performing how they should be, you shouldn't get any difference overall in the group. So when it comes to housing, I think that's always a little bit of a concern to someone. What do we do? What do we often worry sometimes a little bit about those overwintering larvae? And for things like Osteotargia, which Katie mentioned earlier, the way they sort of sit in the abomasal wall until the following season can cause all kinds of problems then when they suddenly emerge, and we get these sort of Osteotargia type disease that we see in cattle. So one of the things that we can do, we talked a little bit about pepsinogen testing. So pepsinogen is what we call a proenzyme. So it's released from the walls of the stomach and it's converted to pepsin in the abomasum. So pepsin is in most mammals, and it's responsible for protein breakdown. And that is not normally present in high quantities in the blood. So if it is in high quantities, it indicates that there's been damage to the stomach wall, and that pepsinogen has ended up in that blood. So we can see that quite clearly on blood sampling from animals as well. Katie mentioned about targeted selective therapy, so I won't go into that in too much detail, but Lucy, can you flip forward for me, please? So lungworms, again, we can base it on clinical signs. So I think some of you may have seen sort of clinical signs of lungworm in your cattle. You'll see that sort of air hunger. You'll hear a lot of animals coughing when they're out grazing. It's quite a definitive sound that you can hear out there. But again, when we get to that point and we know there may be lung damage, it might be too not necessarily too late, as we won't lose animals, but we'll definitely have some production losses there as well. So things we can do. So the Behrman technique is one. So an example this year, I have a herd of cattle. They normally do lungworm vaccinate, but they haven't managed to get it into the first year grazers this year on time. So when they went out, that was a bit of a concern. So we had to be on the lookout for lungworm. And between two visits for fecal egg counts, I noticed a percentage more were coughing than there were the previous time. So what we decided to do was take a muck sample off those animals. So that's that bottom picture you can see. And we did a Behrman. So we basically float the poo there overnight, and all the lungworm larvae swim to the bottom, and we can catch them, and we can actually see them swimming under the microscope. So once we've seen that, we can then decide that we're going to treat those animals. So it's quite a good specificity in that if you see the lungworm there, they've effectively got it. So we treated with an albendazole product, and then I went back 3 weeks later actually. They were still coughing quite significantly. So we took another sample, and we again found lungworm again. So we decided, having had a chat with the farmer about how much they used, product, how it went on, etc. Everything sounded above board, and we realized there's probably some resistance issues there as well. And they did end up having to use an ivermectin-based product, but it did work the second time. So yeah, and the results come quite quickly in sort of 24 hours. You can examine sputum. So I've not done this before, but apparently you can do that quite easily for fairly rapid detection under the microscope. There is ELISAs, and as Katie mentioned earlier, they're not the easiest. They can be quite difficult to interpret. So the problem is you don't get seroconversion until about 4 to 6 weeks post-infection. And then you also find that that antibody level sort of stays quite high for a long period of time. So it's really difficult, I think, if you have an active lungworm problem, to do anything about it using bloods. And again, I'm just going back to post-mortem, it being really important. So I think for farms where you know you have lungworm, vaccination, I would say, is probably pretty paramount there. Lucy, can you flick forward for me, please? So fluke, and again, I drew this. It is a sheep, so the life cycle of a fluke is around 12 weeks. So you can see on the top right there, the sheep ingest these sort of metasaccariae, they're called, off the pasture, which is just a stage of the fluke that we've got there. And that's sometimes when they ingest those, that's where we sometimes see acute fluke. So this is where we get significant damage in acute fluke, where we get large numbers of immature fluke. They migrate through the liver and they cause significant blood loss and sometimes secondary infection. So some of you may have heard of Black's disease, which is a secondary complication, and some of you will be vaccinating against clostridial diseases. But sometimes we can see losses of up to about sort of 10% of the flock here as well with acute fluke. The adult, so the fluke, adult fluke lay eggs, which are released onto the pasture, and then these little miracidium hatch. And then they have, the difference with fluke is it's got this intermediate host of the mud snail. So this is where we talk about pasture and having wet pastures, and people who have wet pastures will probably have seen fluke, definitely either in their herd or their flock. And then the saccariae come out of the snail and it goes back around again. So with the snail, it takes about 6 weeks, and then they can overwinter in the snail as well, and then you get that sort of life cycle resuming again in the summer. And it tends to start this, we tend to see signs sort of late autumn, winter, although that is changing with regards to sort of climate as we're getting warmer, wetter weather. So in terms of trying to keep fluke out, if you have snail habitats but no active fluke, is really important. So using fluke control on animals coming into the herd or into the flock is quite important there. And there's different ways we can approach fluke. So Lucy, if you can flick forward for me. There's different diagnostics available. So I would advise anyone who has either cattle or sheep to get some abattoir reports. So you can get feedback from the abattoir on condemnations or liver fluke history. And I wouldn't say just do it once, or try and do it all the time, make it something that becomes a habit. Your situation with your fluke will change year on year. It'll be very dependent on the weather. And it will give you an idea of fluke infection that's accurate for your farm. So keep checking back on that. There are dairy herds, there's bulk tranquilizers available. Now that establishes the presence of infection. Now with that, again, it's a snapshot view, is not very useful. So if you look at the bottom photo on the right hand side, these are some of the infectious disease reports we do for our farms. Now the green line there is fluke, and the Q1, Q2 is each quarter. So they get an accurate idea of what's going on, and actually monitoring that fluke over time can give us a better idea of the burdens that we're seeing in our animals. You can do for sheep and potentially for cattle as well, you can do individual milk or blood ELISAs, so that's for antibody again. Now that can detect early infection, so sort of 2 to 4 weeks after infection. So that can be really useful if you, after suspected exposure, so to see whether investigation is likely. So doing that sort of late summer, early autumn. I know Dan's done some flock health plans with some suggested testing times on them. Antibodies can persist for up to about 10 weeks after treatment. So doing them randomly may not be quite so helpful, but definitely knowing that you can do them at certain times of year and adding that into your flock plan can be very useful. Liver enzymes again. So liver enzymes are things we look at GGT. I won't try and pronounce it, in GLDH. And now, so GLDH will reflect leakage from damaged or dead liver cells. So that might be elevated in animals with liver damage, and we can usually sort of see that about 6 to 8 weeks post-infection. So again, that's a good time, rather than waiting for that full life cycle when we're looking just for eggs to be produced. We can sort of see whether we think infection is going to be likely and whether or not we need to do something about it earlier on. Fluke egg sedimentation is good. It looks for actual eggs. The pros are that the specificity is good. So if you find an egg, it's likely they've got, well, they will have adult fluke there. But it can miss animals, and it requires a lot of muck as well. So you require about 40 feces from an individual sample, and you're only detecting active infection there as well. Whereas the copper antigen test actually requires a much smaller amount of feces, so somewhere around 5 grams. It's best on individuals, and it is better in sheep than it is in cattle. But about 2 weeks before detection of eggs in the feces, you will be able to do a copper antigen test. But it means looking at everything. So it's farm history, it's time of year, it's weather conditions, etc. And then Lucy, final slide for me. This is a NARDIS parasite forecast. Now I often put these in my health plans for people to look at, and I don't know how many people actually do, but I just wanted to show you how useful they are. So this one, I think that Lucy's got the website for, is this cattle one. There is a SCOPS one as well, so that's SCOPS, that's a sheep one as well. That's pretty good. And if Lucy just goes down slowly, you can see you've got temperature and rainfall, and it's have each month. So October's on this one. You get a weather report, so it'll explain about the weather and what that means for your parasites there as well. And then for liver fluke there, it's really good. It gives you sort of provisional forecasts about what's going on. If you carry on down a bit, Lucy. And then it gives you risk-based areas, so what you can look at there and where you are. And then I think a little bit further down, Lucy, as well, it gives you, oh, I think she's gone. Yeah, and quarantine cheaters, but yeah, it gives you sort of what's going on in the animals, things to look for, and potential tests that you can do as well to give you some sort of accurate diagnostics on there. Thank you.
Great stuff. Thank you, Claire. Thank you, Rob. Thank you, Katie. If anyone's got any questions, I've got some that have popped up as we've been going along there, so I'll start with those, but anyone wants to contribute, feel free, and any other questions, either shout out or pop them on the chat. We did have someone early on. Leslie, you said you'd use some sentinel lambs this year, which is interesting. You've been doing blood tests. I was interested to know, have you been blood testing for fluke antibody? Have you been blood testing for liver enzymes? What blood tests have you done, and how have you incorporated that into your treatment plan?
Hiya. Yeah, I've done monthly antibody tests on the lambs, and I was recommended to do that by Philip Skuse up at Mordon, and it's come back negative so far, which is good. We've done 2, and we'll be doing another one the beginning of November, and we'll see what happens. But we are a fluky farm because we're right down here in the southwest in Devon. We have between 900 and 1,000 mil of rain a year, and this year's been no exception. So it's very valuable for us, and I'm really pleased, because I probably would have given the lambs a flukicide by now, and I haven't had to, which is great, and I'm really pleased about that, and I'm going to do it every year from now on. It costs, obviously, but I think on balance it saves you money in the long run, and it certainly saves building any potential resistance, particularly to triclobendazole, which is what we would have been using at this time of year. So I think it's well worth investigating and doing if you haven't considered it before.
Great stuff. Thank you, Leslie. Really key point there, that what people tend to reach for, if they're not having a targeted approach, they reach for the triclobendazole because, as you all know, it treats fluke down to very immature levels. But with widespread or increasingly widespread resistance to that, we tend to be potentially causing a problem. And another really interesting point you made there, Leslie, that the fluke antibody is only really useful for animals that haven't previously grazed. So in this case, lambs or first season grazing cattle, because it can persist for quite a long time afterwards. And so if you're looking at exposure in a second grazing season, you can't be sure that that's exposure from that grazing season or the previous grazing season. So some really interesting points, and great to hear that it's working in practice. We had a point from Tim. Tim asked about how we can use adults for hoovering up parasites. This is a really practical, really useful application, as everybody's touched upon. Generally, older animals are far more resilient to the majority of parasite infections. Not so much liver fluke and lungworm requires exposure, but certainly sheep and cattle that are reaching adult levels are far less likely to propagate those infections. So whilst they might go on infected pasture, they don't multiply up inside the animal in quite the same way. So we can use dry ewes, for example, pre-tupping that are in good body condition, or we can use our adult dairy cattle to hoover up a lot of these parasites in pastures that we might have considered to carry medium or potentially even higher risk. I don't know if Rob, Claire or Katie have anything to add, or if anyone in the audience is doing similar things, but that's a really practical thing that we can utilize to control parasites whilst maintaining regenerative agriculture principles. So very handy. I'm not sure if anyone's doing that, Rob, but do you have anything to...
Well, yeah, I was going to say, you mentioned sheep systems making use of it, and obviously suckler herds as well naturally make use of that. I was going to ask the audience here, ask the farming community, is anyone doing it with dairy young stock and mixing adults? I don't think I've seen that here in Lancashire, but how is anyone here making use of that? Let's ask the audience. That's either a no or
I think that's something we can work on more, Rob, isn't it? Incorporate into our parasite control plans and things more.
Yeah,
in a dairy capacity as well as the beef and sheep, because it's something that's very good, and with a bit of planning, we can do it really nicely. So Tim, that's probably a little bit generic an answer for you specifically, but certainly in herd health plans and things, thinking about your individual pastures as being low, medium and high risk, and then thinking when you can graze your adults there and have your follow-up young stock, then that can fall into that plan quite nicely. We had another question from Kat, and Kat talked about how we can prevent maggot and fly strike infections whilst also maintaining dung beetle, or more specifically with the products involved, dung flies. And that was when I interrupted Rob onto that. Kat, I don't know if that answered your question at all, or Rob, you have anything to add, but certainly the point seemed to be that if we're able to utilize our ecological control and promote our biodiversity and our variety of species, then actually we find our need for chemical interventions are probably far less. Although that is a bit of a leap of faith when it comes to fly strike. We all know what damage that can do.
Yeah, I'd maybe just add, obviously, yeah, limiting what we talked about today, limiting the use of things that will knock out all the things that will help control flies and maggots. But that will take time to build up those populations of predatory species and of coprophagic species. So there will be a transition where you can't rely on that instantly, like you said, Dan. And we do sell the fly parasites, don't we? Which is one control. I always think that it's not necessarily the only, you can't just rely on that either, but it's a help. A lot of people that use the fly parasites really like them. And I guess if it's sheep, then I know some farmers are selecting breeds with tighter wool to limit the risk from fly strike. And then I guess sacrificial paddocks as well. So if and when you are going to use a product, then trying to keep the dung away from the rest of the farm, and you have one small area for a couple of days, depending on the product, but a couple of days afterwards to, if you like, have the toxic dung kept away from most of the farm. Maybe there's some others in the audience again that might have anything to add to that.
Bruce, do you want to talk about what you're doing on your farm?
Sorry, Claire, what did you say there?
Do you want to talk about what you're doing on your farm? Sorry, I saw your comment that you'd struggled to get to the unmute button.
that was in relation to the last subject there, in terms of hoovering up paddocks. So yeah, we do that with paddocks for young stock. We would have fields that we would have maybe, say this year, we would consider them low burdened, and we would consider those paddocks suitable for putting young stock on next year, because they'll be the most vulnerable. So what we will do now is with maybe cows that we'd be drying off early, we would put them out onto that ground and get them to graze it tight, because those cows will emit less fecal eggs onto the pasture and clean it up, so it'll further clean it. Now, if we go back maybe during the summer, we would probably be using those paddocks for cutting silage off of. So it's to kind of prolong a heavy burden of eggs being emitted onto the pasture. So it's giving the calves the best chance then next spring, maybe next April or May, when they head out to pasture, that there's a low burden. So yeah, that's kind of what we're doing. It is what I suppose is one part of the story. It's another lever to pull.
Brilliant.
Thanks for that.
Because I know we had Tim made the point that clean grazing is hard to achieve in a dairy context, and certainly dairy, beef, sheep, they all have their challenges when talking about clean grazing. But it's nice to hear from Bruce, who's managing to do it, and as he says, protecting the young stock there, which are the really vulnerable ones. Michael, you asked a question about how long to leave pasture. That goes back to the low, medium and high risk, really. If something's been grazed by adults, then it is considered medium risk compared with something that's been grazed with young stock, lambs or calves, which would be far higher risk. So for something to be truly safe from having grazed young stock, it really does need that year break, doesn't it, or some rotation in amongst there, whereas some older stock is going to lower the risk of that.
Dan, can you hear me, Dan?
Yes, yeah.
Does on arable ground, is there any difference in the survivability of any parasites, you know, because they've not got the grass lay to hide in? I don't know.
Are you thinking survivability then within a season, Michael, still?
Yeah.
Yeah,
or across the season, I don't know.
Well, certainly if you're not having anything grazing in there to multiply up that infection level, your survival from season to season is going to be less. But I think your point about survival within a season, that's a harder one to answer. I don't know if anybody in the audience or any of the other vets have any sort of input on that at all. It's a harder one to answer, Michael. Definitely, I think about the survival on the pasture. There's going to be some element of weather contribution to that, but I think the key bit is without a host to multiply up that infection, then your survival is going to be less, and depending on what class of stock you're planning. Because what are you thinking? Tell us about the specific situation you're envisaging.
Just like, for instance, now I've got cover crops that I've grazed once, and they'll regrow a bit through the winter, and I'll probably regraze them again in March. So
what animals did you have on there, Michael? Were they adults or were they young stock?
Yeah, still lambs. Yeah.
Store lambs, yeah. So I guess the lamb thing might be the risk in there as well.
Yeah.
So maybe some early sort of fecal egg counts on there would be quite useful to do.
Yeah.
What are you planning on? What are you planning to graze them on? Are they going back onto cover crops? What?
Yeah. To be honest, having just said what I've just said, thinking about it now, it's probably more likely to be ewes that will go onto it actually. So which may not be a bad thing from what we've just been discussing.
That'll probably be a good thing from that perspective. So again, mixing up those sort of young stock, so we're not constantly having young stock going onto young stock paddocks. I think that's a big mindset change for a lot of people, moving from that sort of, well, these are the sort of calves fields or these are the lambs fields, and that's where they are, and often they're places that are maybe nearer the farm that you can sort of keep a bit of an eye on as well. But changing that up a little bit and switching between sort of older animals and younger animals can certainly make a bit of a difference there.
Yeah, definitely, yeah.
You'd be using your hoovers there, Michael, I think, wouldn't you? That's a really good application of your hoovers. As Claire says, probably still a bit high risk to put young stock on again at the start.
Yeah.
But actually, you will have much lower risk with adult stock, and you'll decrease your risk further down the line with it. So a great use of it.
Yeah, sure. Yeah. Makes sense. Thank you.
Thank you. Leslie made a point about not forgetting other methods of blowfly control as well, traps. And also, I think Rob referred to the parasitic wasp, which can be harder in a grazing situation compared with housing, but still you can put the parasitic wasps around areas of fly breeding and things to help minimize the fly populations. And if you get your wasps in there early, then you'll help prevent the buildup of fly population before it becomes a problem.
There is more coming out about the effects of deltamethrin as well. So your fly products on your dung fauna as well. So things like your fly species that other animals eat. So they've done some work up in Scotland looking at choughs, the bird, and the fact that they obviously eat certain flies, not necessarily nuisance flies that are found in dung pats, but the effect of deltamethrin on those animals seem to be having an effect on sort of chough populations up there as well. So thinking about other animals in our ecosystems is definitely really important.
Cheers, Claire. Will asked a question about how long ML residues persist in manure and slurry. Now, this is Kitey's starring moment, because we were adamant we were going to make sure we were armed with this information. So I'm going to hand you over to the world expert on residues.
The world expert on residues is still looking for the precise paper that talks about it in slurry, but it is variable. Obviously, it slightly depends which of the ivermectins you've used. So there's a difference between ivermectin, moxidectin, and all of those. But pretty much, if it's housed, from what I can find, you're looking at least a couple of months till they can be happy it's not coming through. I am waiting to find a paper that looks at the acidity in slurry, but certainly it will be excreted, and there will still be, because the products are not completely broken down. And even actually, if you use a product called doramectin, the metabolites of it are also still can cause damage. So if you want to be ideal, a good couple of months after they finished excreting before you would be spreading that product. So the other option, obviously, to look at it is whether if you're going to treat less of them, you'll have less and you'll get the dilution effect, and whether that would speed things up. But I wouldn't want to advocate that. But so, yeah, sorry, Dan, still not world expert. I did spend a lot of time looking for a specific paper on this, but the ballpark figure I came up with from everything I found would be about 2 months after they finished excreting, which I say will vary product. Interestingly, if they're at pasture, it's quite variable. So there's some work done with ivermectin, and in the winter when it was cold, when some work they did, it took up to 217 days, but in the summer it could be as low as 7. So climate also really comes into that. And that's also relevant to the overwintering, because there's a lot of work to show actually very cold winters, more larvae overwinter under snow than they do if it's horrible and wet. So that's why Scotland's worse.
I've got a little graph that might help inform this sort of debate, because it is difficult. I'll see if I can share screen. Lucy, is that going to be possible?
Yeah, should be fine.
So can everybody see that?
Yeah.
So basically, this was a study. I guess the main thing to take from this was the sunlight helped degrade ivermectin. So and also having a healthy sort of active soil versus a sterile soil. So they're the two take-home messages for me. If there's a lot of sun and you've got a good healthy soil rather than a degraded soil, then you will get rid of it quicker, if that helps.
Yeah, I think it's where the challenge in the slurry is though, isn't it, Rob? Because it's normally in a slurry tank, and so it's really different. And that's why I can't find the data. And I don't know whether because when I talk to a lot of my European colleagues, they don't seem to use a lot of slurry management in other parts of Europe. So whether the work just hasn't been done, maybe we need to fund it.
Yeah, that sounds like a plan. But I think it's good, that is, because it demonstrates that really these conversations are at the pointy end of the debate, doesn't it? All of this work and the answers don't already exist. But the fact that you guys are prompting those answers hopefully should generate some interest in some work in it. And what I think it demonstrates is actually there is residues there, and so we do have to be careful about our slurry and manure management, and that minimizing treatment through some of these other practices we've talked about tonight and protecting our pasture has to be a good way of going about it. Lungworm vaccinating, Michael, in suckler cattle, I wouldn't necessarily say it's widespread, certainly not so much where we are more dairy. Rob might be able to comment on it from a Lancs perspective, but there's absolutely no reason why that shouldn't be employed. That vaccine works incredibly well because it's a disease that responds very well to immunity. So I don't know, Rob, if you've got a lot of beef clients who are using Husvac or not.
We had a couple of at least one suckler herd in the pilot, and we were watching really closely for lungworm in the young stock. We didn't get it. Now, obviously, they're automatically making use of the whole Hoover effect. So we've wormed them at the end of the season as they've come in. I think he wormed one animal that looked, slightly off. But I think next year he's going to use lungworm vaccination just as a safety net. Yeah.
Yeah, I would say it's spreading into beef. If you look at the map across the UK of lungworm, it's becoming much more geographically everywhere. It used to be very much this west wet coast. I haven't seen a lot of it. We've got beef cows up I'm in Stirling, and we farm up on the Black Isle, but probably quite extensive grazing and a lot of hoovers compared to nicer dairy grazing ground than I would be used to.
We definitely used to consider lungworm a problem of autumn as well, and very seasonal, and we see it all over the grazing season now. It's far bigger an issue than we ever used to see. So definitely something that requires some thought. We had a comment from Penn about ways of treating worm-sick land, some things there that in itself might throw up their own challenges. I'm guessing that this is when resting it isn't an option, or when using those adult hoovers is not an option as well. Has anybody tried any of those things that Penn talks about, heavy liming or growing a thick crop of mustard and ploughing it in, or planting a lot of garlic, or spreading soot? They sound like some very varied approaches to it. Has anybody any experience or tried any of those things? No, I think that's an answer in itself, but maybe something worth looking at more. And I'm just trying to pick out any other questions. Has anybody else got any burning questions on their mind or anything? If not, don't worry. You can always email them over to us, and we can try and follow up, or we can follow them up with some further discussions. Oh, brilliant. So from Tim, we've got walnut for fly control and willow for minerals. I know that was one area we definitely wanted to talk about at some point in the future, is minerals and mineral provision, and some more biological ways of mineral provision. Willow is definitely one of the things on there, so that's interesting. Tim, if you fancy unmuting and telling us a bit about that, feel free.
Can do, if anybody can see. So we've planted 20 or 30 walnut trees round our milking parlour to keep flies down. They give off a pheromone, so we thought we'd experiment. And then we've also put in alleyways for willow trees for salicylic acid, main ingredient in aspirin, trying to look at setting up a paddock for animals that need some TLC. So hopefully that will come to fruition, and we're doing some analysis which should come back next week, I'm hoping.
Brilliant. Cheers, Tim. There's some great work on the benefits of salicylic acid. Absolutely fantastic. There's a licensed product actually now for feeding to calves. And there's also a lot of work done on the benefits of inflammatory feet conditions like digital dermatitis and things. So that's really, really interesting to see.
We have got 5 trailer loads of willow hay. We've harvested it for the first time this year. No idea what we're going to do to it, who we're going to feed it to, but it's all a little bit experimental, and we'll have to go into a shed shortly because the cows will be coming into winter housing very soon.
Brilliant. Well, we look forward to hearing the results of that. We've had some questions about willow tree hay as well, so we've got some live experimenting that can provide the answer.
Hoping it does provide the same tannins. We'll see.
Rob, were you going to make a point? I was just going to ask a question. How you've made the hay then from the willow, Tim? How much are you allowing browsing? Is that a significant part of the system, or not?
Browsing was the main reason for planting it in the first instance. The hay is sort of an offshoot. I wanted to do it as low cost, low maintenance, less intervention as possible, but it started to grow to 12 feet very quickly. So we've pollarded it back to 5 feet, and we've ended up, I thought we'd have 1 trailer load. We've ended up with 5 big trailer loads of willow, from about 1.5 inches in diameter down to just all the leaf material. And we've put it on pallets on trailers to let the air flow. And the one pet goat loves it. So what it's doing for the pet goat, we'll have to see whether it does the same for all the dry cows in the wintertime. So we're aiming to feed it to as many dry cows as we can.
Brilliant. And have you seen a significant difference in the health of the cows, or what impact have you had? And in terms of if they're in the feed, impact has it had on grazing? And I get questions about this from lots of clients, so I'm really interested to hear.
They do graze it. I'd like to think that they would defoliate it an awful lot more and give us lots of benefits. It tends to be the cows that don't want to walk so far, the old Maderas that go in the paddock with access to. So we've got 300 willow trees planted in rows with electric fence either side, and yeah, they will graze it, but I think they were getting a bit strong for it. So. Yeah, it's something we're trying to research whether what we've planted is going to be good or just going to wreck the drains.
We've had a question, Tim, from Rachel, who asks what type of willow you have and when was it that you cut it back?
We harvested it probably 3 weeks ago now, right at the end. So when it was as high dry matter as it could be, before the leaves fell off. We just chainsawed the top bits off at 5 feet, and it varieties wise, it's crack willow, white willow, and then another one that I can't remember the variety, but it came from Bulm as their reed bed style water purifying willow. So very fast growing, and yeah, it's grown phenomenally well in our climate.
Brilliant. Great stuff. Cheers, Tim. We're bang on 9 o'clock, everyone, which is exactly when we were aiming for. So thank you for everyone's contributions, for everyone's continued interest. Inevitably, this will spark more questions and more discussion, which is exactly what we hope. And we hope to keep this going. So thanks to Kaiti for her support, Siva's support. That was critical in keeping this going. To Lucy and her tech, and Rob and Claire and their contributions. As mentioned before, Rob has a video to play us out with. We sort of envisaged it being like the end of a film at the cinema when you have the sort of out